contains the genetic information necessary to make us who we are and help our body function. Starting with the structure, DNA is made up of 2 strands, with several nucleotides on each. These nucleotides themselves each consist of a sugar with 5 carbon molecules, a phosphate group, and any one of 4 nitrogenous bases. Both adenine and guanine are purines, while cytosine and thymine are pyrimidines, although it isn't shown here. What is also unique about DNA is its double helix shape, composed of 2 sugar phosphate backbones or strands that are anti-parallel. In other words, they are oriented in opposite directions, each marked by a 5 or 3 end. The 5 end is represented by a phosphate group, and the 3 end is marked by a sugar molecule, or a corner of a sugar molecule, attached to a hydroxyl group. This characteristic will be important later in the process known as DNA replication. When talking about the cells that make up our body, it is often said that each cell of our body, besides gametes, has the same exact DNA. This is a direct result of DNA replication. The process by which DNA is copied or duplicated. DNA replication begins with the replication fork, this region right here, indicating where the parent strands of DNA are being unwound with the help of several proteins, such as helicases. Helicases belong to the enzyme family, serving to untwist the double helix at the replication forks and separating the original strands. Then, single strand binding proteins take the spotlight as they bind to the unpaired DNA strands. So in this case, single-stranded binding proteins would also be prevalent in this area, where a lot of nucleotides have not bound it yet, or bind it. And they also prevent them from repairing. As the DNA is gradually unwound, nucleotides begin to be added to each of the older strands, starting with a short chain of RNA, usually 5 to 10 nucleotides long. This short chain of RNA is called a primer, and it is synthesized by the enzyme primase, not necessarily DNA primase. At the end of the RNA chain, or the 3' end, enzymes known as DNA polymerases take over and continue the synthesis of DNA by adding nucleotides to the pre-existing chain. So this RNA primer would attach all the way over here, and this would be the beginning of the new strand. So when the new strand begins, the RNA primer is the first molecule to be bounded to the old strand. Afterwards, when enough primers have been bounded or have binded to the old strand, then the role of DNA polymerase III, as shown here, involves adding a DNA nucleotide to an RNA primer. So this could be considered an RNA primer right here, and this would be the DNA nucleotide. And then DNA polymerase III continues to add DNA nucleotides along the growing end of the new strand. Now we move on to leading and lagging strands. So the leading strand is the strand that goes in the direction of 5 to 3. The leading strand moves towards the replication fork, which is right here, while the lagging strand moves away from the replication fork. Finally, you get to what is known as Okazaki fragment. But before that, what should be known is that another difference between leading and lagging strands is that leading strands have only one primer. However, lagging strands that move away from the replication fork require more than one primer. This would be the first primer. Then you have a second and a third and a fourth, or you can have them split up. So 1 primer, 2 primer, 3 primer, 4 primer. After each primer, you have what are known as Okazaki fragments, or short segments of DNA that are attached to the old strand as part of the new strand. However, they are not a complete section of DNA, rather only a portion. And each Okazaki fragment follows its primer. and attach only after or at the 3 end of each primer. So if you have one primer right here, another primer up here, then you have Okazaki fragments or fragment after each primer. So one Okazaki fragment here that could consist of several nucleotides and one down here. Comes the function of DNA polymerase 1, which replaces all the RNA primers with DNA. So let's say this, and so on and so forth until each RNA primer is substituted for a DNA molecule. Then finally, DNA ligase comes along and fuses each of the fragments, or Okazaki fragments, together to finally create a new strand of DNA. So that is how DNA is replicated, with the leading strand that goes towards the replication fork and the lagging strand that follows away from the replication fork.